All right. Yeah. Well, thanks. Thanks to the organisers for putting this symposium together, and to Manuel for giving a very nice introduction to the field that we work in. As he alluded to, there's a bit of overlap between the work we've done, but as we go along, I'll try and highlight some of the aspects that make it complementary, and some of the issues that we think about when we're designing these experiments. So I guess all of you here are aware of the issue of malaria, and that it is an issue in sub-Saharan Africa, but I think recently the widespread deployment of ACTs have made a big difference there. But with resistance emerging and really now quite well established in Southeast Asia, we really need to do something about drugs for the future. And from my perspective, there's a couple of angles I'm looking at this. One is in terms of new drug discovery, and classically that can be a target-based approach, or we've seen that the screening has been very successful with antimalarials, and also the approach of improving existing drugs, like with the OZs, the ozonides, how we worked on the peroxide structure of the artemisinin. Likewise, we need to manage our existing antimalarials better, better dosing, better selection of combinations, and looking out for resistance. And there's obviously lots of talks about that at this meeting, but I really think all of these approaches require, or at least can be optimised by, an understanding of the mechanisms that cause the action of these drugs. And this is where I think metabolomics really has something to offer, because metabolomics, it's part of the systems biology cascade, where people have sort of been using genomics and transcriptomics for a long time, and now proteomics and metabolomics are coming to the fore. Sorry, it should be like that. I think metabolomics is really the most useful of these, because it's close to the phenotype. It allows you to see, okay, this is the phenotype, these are the molecules that we're seeing that are causing this. And Manuel's already sort of been through the principle behind it, that if you have a drug that inhibits an enzyme, you'd expect that when you add that drug to a culture, you'll see an accumulation of the substrate and a depletion of the product. This was an example of a fluornithrin that we did in human African trypanosomes, and it quite specifically showed which enzyme the drug was targeting. But the idea with metabolomics is that we don't just look at one enzyme, we look at the metabolome. So we measure all these molecules, well, not all the molecules, we measure as many as we can, and that depends what analytical platform you're using in terms of what sort of coverage you can get. But in principle, if we can measure a lot of these metabolites and then add a drug to the system, we can see what changes and then try and trace back what the mode of action of that drug is. So I'll go through two examples today. The first has already been introduced, and that's using this approach to screen the malaria box. So I think you're all familiar with what the malaria box is. Many of these compounds had no known mode of action. There's been a lot of work done over the last few years, so we now know a lot more about it. But there's still a lot of unknowns in this box. So we're using metabolomics to try and understand that. The method I sort of worked up for the malaria box screening was to try and increase the throughput a little bit, as Manuel said, in that approach where you purify parasites with magnetic purification. There's a lot of cultures involved, a lot of workup involved. To be honest, if we're analysing a single drug or a few drugs for someone, we often do use that approach. But when I looked at this malaria box and thought there's hundreds of compounds here, I want to streamline this a bit more. So I went for a plate-based approach where we have about 10 microlitres of pellet per well. So the downside of this approach is we didn't purify, so we do have about 90% uninfected red blood cells in there, and of course they're going to give metabolites, which to a large extent are indistinguishable from the parasite metabolites. But thankfully, red blood cells only have a limited number of active pathways in them, so a lot of the metabolites we're seeing are actually from the parasite and not from the host cell. But of course you've got to run those controls so that you know where it's coming from. Extraction, usually using methanol, or sometimes adding some chloroform in there to get some more lipids out of it. Analytical system, primarily using LCMS with helic chromatography for hydrophilic metabolites, and an Orbitrap for the high-res detection is important. With this study, we also used the GCMS approach in parallel, just to get a broader coverage, and that added a few more metabolites to the sorts of things we could see. The data analysis, so the software I developed for this is called Idiom, and basically this takes all the peaks, looks at the masses and identifies as many of those peaks as possible. If we have standards, so we have, like Manuel, about 250 standards that we routinely run, so there's a few hundred metabolites that you know, okay, that's glucose, that's alanine, etc., etc. But the software will also pull out all the other peaks, and all the reproducible peaks can then be analysed to see what's going up, what's going down, and if you think it looks interesting, then you can go back and try and figure out what it is. So we have an automated process to at least annotate most of those. The conditions I settled on were slightly different. So we went with a 1 micromolar drug concentration, given that the IC50s of the malaria box compounds sort of ranged from the 0.1 to the 2 or 3 micromolar range in general. And I went for a 5-hour incubation of trophozoite stage parasites. And when I put in some of the known drugs, sorry, I have a heat map, I don't have the beautiful hexagonal diagrams that Manuel had, but I'll highlight what we have here is clustering of compounds with similar modes of action. So piperacin and chloroquine clustered together, the methylene bluonide and misinin, both quite potent antimalarials that likely act through redox mechanisms, and also the atovaquone was also clustering nicely. The other ones that didn't cluster there, I should mention, are compounds that had an EC50 above the 1 micromolar that I chose. So if it's not potent enough to have activity, we didn't see any effect on the metabolome, not too surprising. But when I looked at atovaquone, so this is a sort of a typical readout. On the y-axis there are all the metabolites, so there's about 500 or so metabolites or putative metabolites that we'll detect. And if it's yellow, it means there's no difference between atovaquone and control, and red is an increased abundance and blue is a decreased abundance. So in this case, you can see there's only a couple of metabolites that have changed, and the ones that increased, as we saw before, are these precursors in the pyrimidine pathway, and the one that decreased was UMP, which is a downstream product of this pathway. So even from an unbiased point of view, just looking at the data, you can immediately see this compound acts in this pathway. Okay, it's not a direct inhibitor of DHODH. We rely on other people's work to confirm that. But it certainly points you in the right direction. Sorry, these things are not... There we go. So when we put in the screen, this is what it looks like. So here we have all the compounds on the x-axis, and all the metabolites that we detect on the y-axis. And overall, you can see there's a lot of yellow, which means a lot of things stayed the same. So it's a pretty well-controlled experiment. In this case, 5 hours wasn't enough to completely kill a lot of these things. And a lot of the interesting things we saw were up in the peptide and nucleotide region. So if I just bring out a few of those for examples, the nucleotides included these pyrimidine precursors that we've seen a few times already now. Yeah, like Manuel, we found a lot of these malaria box compounds were inhibitors of this pathway that cause increased levels of those metabolites. So we confirmed this by testing them in the parasites expressing yeast DHODH. We also did some labelling to show the block of labelling getting through the DHODH enzyme. And actually even with GCMS, which doesn't have as good a coverage as LCMS, you only sort of get 50 to 100 really nice metabolites out. And you can't detect these metabolites using GCMS due to the nature of the analytical technique, but the profile was actually, like the data still clustered together for the compounds that inhibited this pathway. So it had knock-on effects in the mitochondria that we could detect in that profile as well. So as well as the mechanism itself, you do get an overview of which compounds have a similar mechanism of action. Some of the other interesting things up there in the peptide region were these peptides here, which themselves I don't know where they come from a 2-peptide sequence. You can't map it back to a protein. But these are peptides that increase in response to chloroquine and piperacin. And the other compounds in the malaria box that correlated with this, if you look at the structures, you can see they're also quinolines or have similar structures. So it's kind of not too surprising that they might act by the same mechanism of action. If you take a multivariate approach to this data, you can do a principal component analysis. And when we did that, you can see along PC3 here, which is on the y-axis, you get clustering of a few compounds along that axis. What that corresponds to is actually depletion of a range of metabolites, ranging across all different classes. There were 4 compounds in particular. I'll just show you the colours there rather than read out the MMV numbers, but they really caused a significant depletion of a number of these metabolites. And these are basically in the same category that Manuel was talking about before of the cellular homeostasis affecting drugs, that they really deplete a lot of these pathways. The only metabolite that increased for these guys was DHAP, a glycolytic intermediate that's also involved in host cell metabolism. But as well as those 4, I think we had 13 compounds that caused a significant depletion of specific peptides. And artemisinin also caused this same depletion. So clustered in that same group. These peptides that are depleted, based on previous work from Manuel's lab, are mostly mapped back to haemoglobin. And we know that the parasites consume haemoglobin, break it down to produce their amino acids. And so it's not too surprising that artemisinin might inhibit this process. We know artemisinin has a peroxide bond which is activated by heme in the digestive vacuole, so it's perhaps not too surprising that it would inhibit the activity of haemoglobin digestion in this vacuole. The other compounds that had this same effect were the PFATP4 inhibitors, so whether they're directly affecting the food vacuole or whether it's just a secondary effect that there's very fast-acting compounds and their effect on the parasite is to have an inhibition of this pathway, we're not sure. Other thing we're looking at now is also the OZ compounds and trying to dissect their mechanism. Overall, they have a very similar profile to the artemisinins, but we're finding there's an interesting concentration dependence and particularly a time dependence. So it's often worth, if you want to really investigate a compound, looking at the different time points, and we're seeing a different profile for 277 and OZ439 in terms of their onset of action. So Carlo's the one leading this work at the moment, and actually there is a new position if anyone wants to come to Australia for a postdoc. Come and see me after. I don't know why you'd want to leave America right now, but that's up to you. A couple other interesting things we got out of this, which is sort of away from the existing drugs. This compound here, methyl erythritol pyrophosphate, was significantly depleted in the 4 of the potent compounds I showed you before, but also this D6, which is just the plate position. So this metabolite is in the isoprenoid precursor biosynthesis pathway, and actually, so it indicates that this drug must be inhibiting this pathway. And since doing this experiment, there's now been 3 other publications come out using completely independent means to show that this drug that I've called DO6 is inhibiting ISPD, an enzyme in this pathway. So that's been nicely validated by a number of other groups. One of the more interesting metabolites that came out of the untargeted screen was traumatic acid. I had to do a bit of research to find out if it was a real metabolite first. But yes, it is, and it was depleted significantly by this CO7. And it actually has already been shown to be present in Plasmodium. So Joanna Daly's group nicely showed the presence of this linolenic acid pathway in Plasmodium, which previously was known to be in plants. And traumatic acid is one of the end products of this pathway. So it's likely that this compound C7 is inhibiting something in that pathway. Further work will need to be done to nail down the exact target. So yeah, like Manuel, we managed to classify a lot of the compounds that we tested into mechanisms similar to existing drugs, also found a few new targets, and there's still a lot of compounds there that we didn't identify a metabolic perturbation, so as was alluded to, they might have other effects or we may need to treat for longer or at higher doses. So in my remaining time, I'll just introduce a bit of new data we've been looking at, which is to do with understanding resistance. And I think most people are aware that artemisinin resistance is a big deal at the moment. The clinical phenotype's well described as this prolonged parasite clearance time. In vitro, we see it as a decreased efficacy in ring stages. It's known to be associated with K13, the Kelch mutations. But I think there's certainly scope for improved biomarkers for the artemisinin resistance, and certainly to understand the mechanism in more detail. So metabolomics might have something to offer here. For this work, we used the strains produced in David Fiddick's lab, where we proved the involvement of K13 in artemisinin resistance. So we had 2 resistant lines with the 539 and the 580 mutations and compared them to the sensitive line that's on an isogenic background. We also looked at some field isolates from Palin that we got off the Antilles lab as well to see is there an association between the K13 mutations and the biochemistry of the parasite that might be linked to the mechanism of artemisinin resistance. So rather than just do metabolomics, we thought we'd better take a bit more of a global view. So we did proteomics and peptidomics as well on these samples. And Giselle, a postdoc in the lab, set up this method to sort of get as much as we can out of a sample. I will say this is not done in a 96 well plate. There was, I think, 3 or 4 flasks per sample to make sure we had enough material to see everything. So just going over the metabolomics data. In the principal component analysis, so an unbiased look at the data, there wasn't a huge difference in the metabolism of these three strains when we're comparing the isogenic strains of the two resistant lines and the sensitive line, which is perhaps not too surprising for a single point mutation. If we do a supervised analysis, so a PLSDA, where we actually look for what differentiates these three groups, we could see there are some metabolites that differentiate the strains that we tested. And we're still going through the process of identifying all these changes. But we can see that there's an increased glutathione level in the resistant strains, in both the resistant strains, to a small extent. And there's a depletion in some peptide and some amino acid metabolites. So what we thought is we'll have a look at the peptidomics data because peptides seem to be interesting. I won't go through the method now, you can ask later. What we found, so this is just analysing the endogenous peptides, not the proteins, not the intact proteins. And of course we'll identify a lot of haemoglobin peptides. And actually the haemoglobin peptides in general were lower in abundance in the resistant strains than in the sensitive strains. So there may be a decrease in haemoglobin metabolism, whether it's related to a decrease in or sort of a dormancy type phenotype. We haven't sort of put those connections together yet, but it's quite possible. Then we had a look at the proteomics as well, see if there's anything else different. We first looked in ring stages. There's a handful of proteins here that are significantly different, although the ring stage experiment was particularly difficult to get enough material out and to get reproducible data. And so the proteins were in a few of the pathways that are quite common. I won't go into that in detail. But we then did it on trophozoites because then we could get a lot more material and get a much more accurate analysis of the proteins in there. And we saw proteins from a number of different pathways. It was worth looking at the two strains. In the R2 that I've called here, which is a 580 mutant, we saw a few more differences, so some differences in the hemoglobin degradation pathway and some protein trafficking. Whereas in the 539 mutant, there were very few differences that were observed. Looking at the field strains, we see a few more differences. They're not on an isogenic background, so it's perhaps not too surprising. We see some differences in drug transporters and some other membrane components that may have been selected under different pressures in the field. But at the end of this, across, I think 13 or 14 independent experiments, there was only one protein that was significantly different across all of these, and that was actually K13 itself. So in the ring stages and in each of the troph stages, you see a roughly twofold downregulation of K13 protein in these K13 mutant lines. Of course, with omics data, you don't believe it until you've seen the raw data. So we went in and we looked, and there's quite a number of peptides detected from both the C-terminal and N-terminal end. And this is all dimethyl labelled data, so we can see here the reproducible depletion in the resistant strains. So just to summarise that work, we've seen that the K13 mutations associated with a depletion of the K13 protein, not a complete loss, but a downregulation. Also decreases hemoglobin-derived peptides, and there's a small increase in glutathione levels. What this means in terms of mechanism, of course, this is an observational study, but there could be an involvement of oxidative stress response, unfolded protein response, or an effect of hemoglobin degradation. So hopefully this will provide a new option for a biomarker for artemisinin resistance. Of course, this is just a couple of strains in the lab. We now have to expand this out to a lot more strains from the field to see if this is something reproducible beyond these first exploratory experiments. So with that, I'd like to thank the lab, particularly Giselle and Carlo, and also collaborators, so Malcolm and Stuart and Leanne at the Bio21 Institute, and also David Fiddick as well. And for anyone that's really keen on metabolomics, the Metabolomics Conference next year is going to be in Brisbane at the end of June, so I hope I'll see some of you there.
Okay, very nice work. I had a technical question, actually, about the level of Kelch13 in resistant and sensitive parasites. So in the original analysis by Mark et al., they don't see a difference in the level of transcript. And in Western blots, when we've looked, including in David's CAM line that we have, we don't see a level any change in the level of protein.
Yeah, I mean, we haven't done a Western yet because we don't have a good antibody for it, so I wasn't aware of that. But yeah, as I said, we've done this sort of 13 times and get a reproducible twofold
and
decrease.
actually we've done proteomics.
So,
not quite in the way that you've done it. We've done it on a particular subcellular component that we think is relevant to resistance, and we don't see any difference in the level of Kelch peptides that come back between resistant and sensitive.
No, this was quite reproducible with a lot of peptides. This is a saponin-lysed parasite we did the proteomics on. So it's a whole parasite,
our fractionation is even further. So it's actually an immunoproteome.
Okay, so that's quite different.
Right, it is quite different, but we don't actually see a difference.
Okay, that's interesting.
At the level of Western or peptide levels. So.
Okay.
Okay, well, I guess it's a hung jury, so I will say that our proteomic analysis does show a decrease, actually, in K13 in the mutants. So, who knows? I guess it's very, but at least the data we have, these are whole cell saponin-lysed preps. But, and we don't know. I don't know if you have looked into whether this is a reflection of increased turnover, or are there modifications to K13 itself that are leading to enhanced degradation. I'd be curious to know if you've looked into that. And secondly, have you... okay, well, you think about that first question, I'll think about what the second question was in a second.
Well, for the first question, as was mentioned in the transcriptomics data that's published, we don't see changes in transcript levels, so it leads me to think maybe it's a turnover issue, but we haven't...
You haven't looked for post-translational modifications of K13 itself?
We haven't observed any, but we haven't tried to enrich for them, which you often need to do to find these things.
And have you run experiments where you pulse with, DHA, very short-term pulsing, for example, to see if that gives a greater signal either in proteomics or metabolomics?
Yeah, so they're underway. So yes, it does give a greater signal, but I don't have reproducible data yet to, I like to repeat things a lot of times before we show it.
Thanks.
Had a question about the PCA analysis on initially between the Kelch 13 mutant and the non-one. You said there was not much of a difference on the PCA of the metabolites. Although, was that trophozoites or rings that you showed that for?
That's trophozoites.
Right. And then if you test rings at 1 micromolar, so one, it'd be more interesting to look at rings. You might see a difference. But then you're probably going to have to go higher because 1 micromolar is closer to 1 or 2 times an IC50. So you probably have to push your drug concentration higher for that.
Yeah, and this is all unchallenged, so this is not drug treated. First, we have to treat it, we have to look at rings, and as you say, probably a high concentration. I think it's worth doing the concentration range. So, yeah, this is just the initial untreated.
So, so the first PCA was more trophozoites and not...
not rings.
Yeah.
Yeah.
Okay.